Start.
Hello students, we are back for the study of the topics in the industrial microbiology. The previous sessions we have looked into the basics of fermentation industry, the crude media formulations, then the agitations. Agitators we'll be discussing separately, impellers and all in the coming class. Then we have also studied about the heat temperature regulation, monitoring and regulation processes, and oxygen transfer rates. Okay, so today's session we will learn about the types of bioreactors. Actually, though your syllabus tells about some specific ones like photobioreactor and you have the one of the batch bioreactors like the various types we have. Okay, I'll be discussing that here, but don't be specific about fluidized bed or bubble cap, bubble column bioreactors and all. They are of course in your syllabus, but we have to go from the basics. When you go, we have to differentiate it. Okay, let's understand about it. So if a question is asked like related to this, explain the types of bioreactors, we can definitely start with batch bioreactors, which includes the photobioreactors that we will deal it separately. They are called specialized bioreactors. Okay, so I think you got the point what I'm trying to say. We will be classifying bioreactors into batch and continuous types. Okay, so we will be learning how they have designed from the traditional to the present ones. We will be discussing about that in the topic of today. Okay, materials, pictures, we will go with those studies as usual. Okay, so as I was telling you, we will be studying about the batch fermentation process. Okay, so we know that bioreactors are the ones which are going to help us in producing various products. Okay, so we have to provide them with the ingredients like the diluted molasses, whey, and sometimes hydrolyzed starches. Okay, what does the meaning? So once you give the media, you have to add the inoculum. Okay, so an axenic culture means a pure culture of the microbe which is actually going for inoculum development. There can be a class on inoculum development. Okay, so you have to, that is called as upstreaming process. Inoculum development is an important part of the industry. Please keep this in mind. There can be a question over that also. The broth is adjusted with a specific pH and temperature with supplements. Okay, and the medium is taken in the bioreactor and it is steam sterilized. The inoculum development and scaling up is a very important part with the crude medias. Okay, the final scaling up medium. The final scaling up is very important. Okay, so that it will go to the main bioreactors for the production. So at every point of time, there is a requirement of monitoring the pH, temperature, and quality control, and all happens every step. So what do we monitor generally? Contaminations, inoculum concentrations, desirable products, and undesirable products. Please remember, desirable products should be always higher than the undesirable products. You cannot avoid them. They will be produced during the fermentation process, but it has to be monitored. So we have to concentrate that it should be higher than this. Okay, yeah. The incubation period, like it depends upon the type of the product anyway. Harvesting is an important part. Okay, after incubation, we have to go for harvesting and then go for the downstreaming process. Okay, and please keep a note of this. Whenever you talk about batch fermenter, the fermenter is cleaned with, after harvesting, again it's completely cleaned, sterilized, and then goes for the next batch of production to start. That means each fermentation is a discontinuous process divided into many batches. So every batch is to be monitored. Every batch is different. That's what it means. Accordingly to, if you say, talk about this, all the fermentation industries earlier were of batch only because there was no any innovations like continuous. Okay, so keep this in mind that a batch batch fermentation process is a very, very basic, very, very traditional type of system. Okay, so I will be talking about this various types shortly. What is the advantages of the batch? Economical, more conventional. Okay, there's no too many monitoring systems. Disadvantages is you have no specific time bound process. It's like, there can be low productivity. One has to decide whether to harvest it because there can be some discrepancies in the production. Okay, difficult in maintaining the inoculum concentrations. Okay, what are the different types of batch fermenters? Coming to one of the first one, Asahi tanks. Asahi tanks is typical bioreactor with the cooling jackets and agitators and all like this. This is an Asahi tank, a very basic model of a bioreactor, which all of you are familiar with. Coming to unit tank is a conventional tank with inner wall made of tiles, no inlet and outlet pipes, but works simple by loading and by suction. So see here, this is a simple unit tank where earlier people used to go for production of alcohol, mass production of alcohol, but there was no any specific equipment. It's just a tank in which the fermentation would occur. It's not a it means it cannot be consumed in the sense. Alcohol, whatever you can produce by molasses from here, can be used by distillation process, not for consumption, not wine, etc. That's a basic unit tank. Okay, small cylindrical conical tanks. Here there's no agitator, it's a conventional air current given, vigorous fermentation occurs here. See this picture, you can understand this. This is a cylindrical conical tank. So we call it a cylindrical conical tank. Okay, there's a glass viewer. So this is the harvesting line. So here is the spargers. Okay, there's no any agitators here, and fermentation goes here. So you can see that this will be the ground floor in the industry, first floor, second floor, third floor. This is how a bioreactor will be installed. So only the topmost floor where I'm showing the pencil here, above that only is the top floor. So any person who is here in the top floor will be seen through the glass viewer, which is seen here. Okay, this is a glass viewer. Through the glass viewer, he will see the fermentation process and all. Okay, anyway, this is for monitoring. Remaining all are automatic systems. This is a modern bioreactor, not a traditional like the other ones. Advantage of this, we can produce, we can save the power. There is no agitators, impellers and all. It's fine. And the vessel is, the coating fabrication is very smooth. Easily everything is drained off. There is no any provision of cleaning difficulties and all. So it's a very good one. Disadvantage is difficulty in foam control. More hydrostatic pressure will be there. So we don't know what the condition of cells sometimes that can affect the cells. Okay, that's about the cylindrical conical tanks. And here you find another next one is the spheroconical tanks. This bioreactor is having a spherical shape in this means in the sense bottom it's conical, and as it goes it becomes broader. I'll show you the picture first, you'll follow. This is a basic idea of that. So you have what you see here, large headspace will be present in this. Agitator, everything is there as usual. So imagine this is the fermenter, huge tank. It is quite, it can hold a high amount of media. Here there is a wide space, no? So cooling will be very fast. Temperature regulation is more easy. There is no need of jackets, all those things. Headspace is huge, cost effective. Okay, it maintains the temperature very well. It sends high carbon dioxide concentration, no pressure. In the previous one, hydrostatic pressure will destroy the cells and all here. In this case, it is not that much. It easily controls the foam also. So foam control, hydrostatic pressure, everything is a very positive aspect of the spheroconical tanks. Okay, so it's one of the recommended batch type of bioreactors which are used in many of the industries. Okay, I mean to say it is something very similar to this. I can just show you a picture. Okay, this is imagine like this it should be. Okay, so this is a spheroconical tank. So what you're seeing in green color is a harvest line. Okay, top portion, and the middle portion is quite huge. Just to understand, just like a top shaped condition, but imagine it will be huge. Gallons of media can be holded in this. Okay. Continuous fermentation process is a very advantageous one compared to the batch fermentation. In batch fermentation, you feed the medium, you inoculate the cells, you harvest the product, and the next batch is with cleaning and again adding fresh medium, right? Continuous fermentation is a different setup where you need to cultivate the cells, harvest the medium, the product, and continuously again add the inoculum and the media. So something you'll understand the differences as we go ahead. So there is no intermittent complete harvesting of the media. Partial harvesting of the media will happen. Okay. So The microbes do not reach the stationary phase since end products do not accumulate, does not inhibit or kill the cells. The cells are said to be in steady state and the condition of homeostasis. So what do you understand here? So the cells are remaining in one phase. Though we have synchronous culture, it is different topic. I'm not going into that one now. Let me discuss that later or sometime. Let us, if there is anyway topics related to that. Okay, continuous fermentation is something where a process occurs. You are going to add the inoculum and bring in the same bioreactor. Fermentation is going on. There is no immediate harvesting. You get the product also intermittently. So it's a very advantageous one. We'll go with the diagrams. We'll follow it up a little more better. What are the characteristics of this? Fermentation medium is taken in the bioreactor. Inoculum is added. That's the first two things which we discussed. Okay, add specific incubation. Bring a specific incubation. Fresh medium is added at fixed rate with some specific supplements. Okay, the parameters are aeration, pH, all maintained. Nutrient medium is added simultaneously and drained off means it is used off. The valuable product of fermentation with the maximum concentration is drawn with the receiving vessels for the downstreaming. In the sense, your harvesting will be high. Talking about the same, let me continue with this. The inoculum cell density in the continuous fermentation can be monitored by two methods. One is chemostat and one is turbidostat. It's a very basic study. Chemostat is the flow rate of the fresh medium and the harvest. They are maintained constant with the help of flow rate regulators. Okay, how to make you understand this? If you see this picture, this is a bioreactor. So here is the inflow of the fresh nutrient medium. So don't think that complete medium is like, all ingredients are added. Some basic media with some supplements will be there, vitamins like that. So that this will have a control over the growth, turbidity. If you give the specific nutrient only, this will be able to grow. You're keeping them in a starvation. You're keeping them in a stationary phase. Okay. You provide them, meanwhile product will be produced. You, as the flow rate, whatever is here, and increases, and that will be released and collected. Okay, to make you understand, amount of medium added is equal to amount of medium harvested. So this is called as flow rate regulator. To make it more advanced, you have the turbidostat. In turbidostat, what happens is there's a device which is very sensitive, photoelectric device. Okay, it will measure the turbidity, which is connected to the fermentation tank. Automatically fresh medium is added to the bioreactor. Okay, so that the equal amount of medium is harvested. How to make you understand this? See here, this is the similar diagram which will tell you. Here is the tank which adds the medium. Here is the photoelectric photosensitive cell. Photosensitive cell is here, and this is a light source. As light passes, based on the density, it will check the turbidity. That is going to be monitored by the amplifier, photoelectric cell amplifier. Then it's going to be helping in bringing a process where it will regulate how much to add here, so that these cells will be always in the phase of what? Starvation. Okay, logarithmic phase. Of course, I've not shown the draw line. From here you can show, from bottom you can show harvest line. Okay, so you can always use a similar setup like what I've shown here, where it will flow. So amount of media fallen, amount of media come out. So turbidostat is also one of the important technologies which are very useful in the industry. Don't think that you're using this only for studying about growth curve and all. Not like that. You must try to implement whatever you study in the growth chapter for bacteria or any organism by these techniques. Of course, we all know that turbidostats are more suitable for bacterial cultivations, not very much for the fungal cultivation, because their turbidity is very high. Density is very high in fungi. Okay, so that's one of the informations what we can rather gain. There are different types of continuous fermentations. So what you see here is one of them is single stage continuous, recycle continuous, multistage continuous, which is again classified into simple chain and complex chain. You need not worry about it. We'll understand it as we go ahead. One of them is called single stage continuous. Very simple. This is a bioreactor which is called as continuous stirred tank bioreactor. A single fermenter is inoculated with the medium, and operation is going, continuous operation will be going on, balancing the input and output of the fermentation medium. That's all. Nothing much great about it. So you just have to bring a continuous fermentation process. just like your batch. It's very similar to the batch, but it's a beginner of continuous because it's showing addition of fresh medium and all. You call it as continuous. That's all. Okay, so similar to the first one what you have studied. Okay, coming to the second one, that is the single stage recycle. Very important here. As the fermentation proceeds, the harvested medium containing the cells are recycled back to the bioreactor. I think you got the point. By this, continuous fermentation can be made more faster. So we learn a technology, but we should also understand why it has been used. What is the meaning of this? No, let's see the diagram here. In the diagram, when you see, I'm not labeled anything, please don't mind. This is a medium already taken. It's a bioreactor. You can add the fresh medium from here. Okay, that's a condition. What does it mean? Single stage recycle means in singles bioreactor, we recycle the cells back into the same. So what's the meaning of this? You are adding the cells back into the same medium. So some will be growing here, some new fresh cells will be added here, so that the fermentation, what's the main intention? The fermentation process should go much more faster. In this, you would also harvest. And then the remaining cells are recycled here. Okay, that is called as single stage recycle process. This is a multi-stage continuous. Multi-stage continuous means it involves more than one bioreactor which are operated in a series. Even though it is multi-stage, all the bioreactors are able to maintain the parameters, very important. pH, temperature, QC, quality control, all those things are maintained. Multi-stage, please remember that it is more than one bioreactor. Okay, many. So here I've just tried to show you a very simple picture, easy to follow. So it's a cascade of bioreactors. In Japan, they have a cascade of bioreactors where they don't have, there's no any point of time, there's no harvest. Every day harvesting will happen. Why? Because there is a inoculum which is developed here, it goes here. When it's added here, the media which is present here will be activated by the cells. Whether it's initial lag phase also, it is taking it up. Here, what is the meaning of this? Why do you do this? See, here you are pitching. Pitching means adding the inoculum. Then it should come to a stage where it is all having something like some amount of turbidity will develop. It will take some time. Like you get the cells. So here what you do, in this simple chain, you would pump from cells from here, right? So already the cells are in the, maybe in the logarithmic phase also. Then the growth rate will be faster. So what are we doing here? We are saving the time. That's more important in simple chain. Saving the time is enough? No. By this, you can do everyday harvesting. You need not wait like batch process. When batch process, you have to wait. See, in the simple chain, see, cells are going. This is just to understand that there is some amount of processing like centrifugation, cell density, volume is going to be added like that. Okay. So this is also similar setup, whatever you are seeing here. Simple chain, see? So it's like this. So cells from the previous bioreactor goes to the fresh one, making this grow, making this time of incubation shorter so that we can harvest sooner. Instead of 20 days, can harvest for within 12 days. Like that. Okay. So that is one thing we have to understand, simple chain inflow. Complex chain means like you have a bioreactor. So I'll read it out first for you. The cells from second bioreactor is brought back to first bioreactor so that the harvesting the product is more fast in the first bioreactor. So just imagine a picture here. First bioreactor, second bioreactor. You are pumping the cells from here. So already you have done this process of simple chain. You are bringing a complex chain, making the time more faster to harvest by pumping some cells back into this. So by this what happens is your maximum media is utilized, nutrients are utilized, cell growth will be increased. You can at one point of time can harvest almost half more than whatever you harvested in gaps. Okay. By the time this bioreactor will get ready, within a few days this will get ready. That time this is ready for what? Harvesting complete. Though some amount of cells will be going here, this is ready for harvesting. By this what happens is you can monitor them, this bioreactor and all. Okay. So at one point of time, maybe it's gone to the fourth or fifth. This will be empty totally. This will be like, cleaning, goes for cleaning. All these valves are closed and it goes for cleaning. Okay. So same way, as you go ahead, this might have gone for cleaning. I'll come back to this picture here, almost like this. So you have series, no? By the time it reaches this stage, these two would have undergone harvesting and cleaning. Just imagine like that. So it goes this way. Okay. So you find that there is a continuous, what you say, a process where the simple chain and the complex chain will be seen with the process wherein the harvesting will be more better in this mechanism. Okay, so we can consider that the first bioreactor is in the end process. By the time the fifth bioreactor means this will be in the process of growth, whereas the fourth one or fifth one will be in the initial stages. By the time this will finish with harvesting, like that. Okay, so what is the meaning of the whole cascade process is you can harvest more amount of the product, and you can what you say, you can always run the fermentation industry continuously without any break. That's one of the best advantages of a continuous fermentation process. If I want to repeat it, I'll just repeat it in a very brief way once again, because you have to understand simple and complex. Simple chain means it's a series of bioreactors like here. Whatever is grown here after 2 days, give me an example here. After 2 days, he is pushed into this tank, which is the first day for the tank. So this is in the from second day it has come to this. Okay, so here the cells would have already gone to the logarithmic phase, many of the cells. So it will grow faster here. Your product will be produced much faster here. And after 2 days, you're going to push this cells to this. So this will be ready. Okay, so these all are in a very acceleratory form, very fast producers. Okay, so this tank, whatever is there, you can harvest it completely after some stage, and maybe 4th day or 6th day, complete harvesting. This tank is empty now. Okay, you can understand like that. So what have you done by this mechanism? You have saved the time. But the same thing if you make it complex, imagine same thing second day, 4th day. So let's say your 6th day is a final harvest here. If you do this complex chain, the cells would have come from here also back, so that the fermentation will go faster. Maximum utilization of nutrients happen. Harvesting can become 3rd day also or 4th day also. Okay, so reducing the time of harvest by the whole process. This continues a similar way. This is called as complex chain. Okay, so please remember, it is different from simple chain. Cells from the second bioreactor is brought back to first bioreactor, so that harvesting the product is more fast in the first bioreactor. So students, with this idea, maybe you would have understood what is the difference between a batch and a continuous. What's the advantage of a continuous? In a continuous process, though we have monitoring to be done for a cascade of bioreactors, 8 to 10 bioreactors sometimes. It's a very huge operation where every bioreactor has to be maintained. But there are equally manpower is required and automatic systems are required. Whatever energy you put for a single bioreactor, we may have to put for all. But remember,